All right, so thank you. My name is Sean Prosser. I co-lead the innovation unit at the Center for Biodiversity Genomics in Guelph, Ontario, Canada, along with Robin Floyd. And I would just like to also acknowledge the other members of our team, Saida, Amina, and Paul. Everyone contributed in one way or another to the work I'm going to show you today. So before I talk about how to sequence amplicons at a large scale, I'm first going to quickly give a few examples of why you might want to do that. So this graph shows the number of amplicon-based human genetic tests available in the United States every year. So by 2022, there are about 35,000 different genetic tests available, all of which require amplicon sequencing. And as you can see, the number of tests has been going up every year, so there is going to be an increased demand for amplicon sequencing because of that. But you will probably also notice that it plateaued a little bit in 2020, and there's a good reason for that. As you all probably remember, the world kind of shut down then during the coronavirus pandemic. So not only was there a shortage of lab supplies, but a lot of labs directed their attention towards combating the virus. Which leads me into my second example of amplicon-based testing. So between February 2020 and February 2022, there were over 2 billion COVID-19 tests performed. And millions of those were positive. So the spike gene of the virus was amplified, but it was not sequenced. So you can imagine if we were able to sequence all of those amplicons, we would be able to reconstruct basically the life history of the virus as it spread around the world and evolved and mutated into different strains. So that kind of data would be incredibly useful, especially when helping to prepare for the next pandemic. And then my final example here is more in my area of science, biodiversity science. So there are an estimated 20 million species on Earth, multicellular species, and of those we've really only categorized about 10% of them over the last roughly 300 years. So there is a lot more work to do, and we don't actually have a lot of time to do all that. It's going to take forever at our current pace, but fortunately we can use DNA barcoding to speed up the registry of all species on Earth. The premise of DNA barcoding is that you can identify a species without sequencing the entire genome. In fact, you don't even have to sequence a gene. You can sequence a small 658 base pair part of the CO1 gene, which is found in the mitochondrial genome of most multicellular animals. It's about 1/1,000,000th the size of an average animal genome. And I should point out that most of the animal species on Earth are actually insects that have comparatively smaller genomes. So that's why the average is only 600 megabases. Vertebrates obviously have much bigger genomes than that. So in order to use DNA barcoding, what we need is really a global barcode reference library for all species on Earth. And that was kind of our main goal at the Center for Biodiversity Genomics in Ontario, Canada. So we've been working on that since about 2008. This graph shows our barcode or sequencing output every year. So currently we sequence a little over 3 million samples per year. I'm just going to take a 30-second detour, though, before we get back to this, to say if any of you live or know someone who lives in any of these dark areas on the map, these are areas where we are lacking barcode coverage and we could really use your help. If you think you might be interested in deploying an insect trap, it's a passive trap, and kind of monitoring it for a period of time, please Google bioscan.life or come find me after this session, or I've also left this map on the bulletin board out in the atrium with more information there. You can go take a picture of it. But these traps are very easy to set up. We have school children doing it in Canada to survey the biodiversity of their schoolyards, and the kids love it, so it's very easy to do. So if you think you might be able to help or be interested in contributing, please get in touch through that hashtag, or please come see me or find that bulletin board post. All right, getting back to it, now I'm going to talk about how we actually sequence amplicons at a large scale. I like to start with this quote, nearly a century-old quote: "No method of advancing science is so productive as the development of new and more powerful instruments and methods of research." This is especially true for sequencing platforms. So from about 2002 to 2017, we were kind of stuck with Sanger sequencing, where we could sequence 95 samples at a time at a cost of about $10 per sample. However, in 2017, we adopted the PacBio SQL sequencer, which allowed us to sequence 9,120 samples at a time at a cost of only 25 cents per sample. So this was a major breakthrough for us. And right now, our Oxford Nanopore hosts are probably getting ready to cut my mic for saying good things about PacBio, but give me one more slide, I promise. In 2024, we started migrating over to the Oxford Nanopore MinION sequencer. The reason we did this at this time was because of the release of the 10.4.1 chemistry, which led to a drastic improvement in sequence quality, and all of a sudden we could now leverage this technology to produce reference-grade barcode sequences. Using this MinION platform, we can sequence up to 100,000 samples at once at a cost of 1 cent per specimen. So this was, again, another giant leap forward for us in our goals. There are sort of two main advantages to the Nanopore system. The first one is that it's very inexpensive. So to actually buy the sequencer and to use it is very inexpensive compared to the other platforms. And the other advantage is it's very flexible. So you can sequence really any number of samples, but we sequence in batches of either a little over 2,000, which corresponds to 24 96-well plates, for about 10 cents per sample. We can also sequence a little over 9,000 samples, which corresponds to 96 96-well plates, for the same price using the MinION flow cell as opposed to the first one, which uses the Flongle. Or we can go up to a little over 100,000 samples at a cost of 1 cent per sample using the MinION flow cell, although we do have to employ ONT's native barcodes to achieve that. So this last scale, this level, 100,000, that's the one I'm going to be talking about. So whether or not you're doing 2 samples or 100,000 or 1,000,000, you have to tag your samples, because NGS platforms basically just give you a giant pile of sequence reads at the end, and you need a way to map those back to your samples. So we do that using these molecular sequence tags, little short pieces of DNA that are unique sequences to each of your samples. Traditionally, people would just ligate these onto their samples after they amplified them, but you can actually just attach these samples sequences tags right to your primers, and then they get basically added during PCR. So this is the much more efficient way to do it. In this example, if you have 95 samples, you would have to use 95 different tags. But there's a much more efficient way to do this, and that's by using 2 tags, one on each of your primers. By utilizing this dual tagging system, you no longer need 96 different tags to tag an entire plate. You can just use 8 tags on your forward primer distributed across the rows, and 12 tags on your reverse primers distributed across the columns, and that will be enough to give you 96 unique combinations. So let's say you do this 3 times, and you generate 3 pools of tagged amplicons. You could then sequence those directly by using 3 different flow cells, or you could use ONT's native barcodes and ligate a different native barcode onto each of those pools. And then you can pool those together and basically sequence your pools of pools. So this is sort of the general way you can increase the number of samples drastically that you can sequence at any one time. So this is how we sequenced 100,000 samples at once. So we have 96 forward primers and 96 reverse primers tagged with unique tags. So when we amplify using that schema, that gives us 9,120 uniquely tagged samples that we then pool into a single tube. If we do that 11 times, that gives us a little over 100,000 specimens. And then it's pretty simple matter then just simply to ligate on 11 different native barcodes during library prep, after which point we can pool them into one final library and sequence on the MinION. All of these 100,000 samples that we sequenced were previously sequenced on the sequel, so we already know what their barcode sequence should be. This just shows a Venn diagram of the success of those 100,000 samples, and as you can see, it was very comparable between the 2 platforms, and in fact, ONT platform actually outperformed the sequel in this case. When we look at the actual sequence composition of those sequences, they were nearly identical. So on these phylogenetic trees here, the green are the known sequel barcode sequences, and the black are their counterpart sequences. So you can see that they are pretty much identical in those trees, and in fact, over 90% of the samples were completely identical. And the other 10% had very few nucleotide differences between the 2 platforms, which was insignificant when it comes to barcode identifications. So we were very happy to see these results. It basically showed that, yes, nanopore sequencing can be used to generate really high quality barcode sequences and help us build that global reference library. Now, in the innovation unit, we like to torture ourselves. So once we were done with 100,000, we asked the question, can we sequence 1,000,000 samples at once? And we thought, well, technically we can. We have a PromethION sequencer, so mathematically it should work out. And if we were able to do that, that would bring the cost down to 1/10 of a cent per sample. But you'd have to be crazy to try that. But we are, so we did. So we also have access to millions of samples because we have all these pools of 9,120 amplicons from our barcoding reference library building efforts. So we thought, well, let's just take 110 of those pools. That's a little over 1 million sequences. We'll ligate on native barcodes and sequence them, the same as the 100,000 project. Unfortunately, ONT only makes 96 native barcodes. And we thought, well, we'll just do 96. But then that wasn't quite 1 million, and it drove our OCD crazy. We're like, no, we got to figure out a way to hit 1 million. So we came up with this idea of basically doing a dual native barcode ligation approach. We weren't sure if it was going to work, but basically on the left we have standard library prep where you just basically ligate on your native barcode if you're going to use it, ligate on your sequencing adapter, and then sequence. On the right we have our dual approach where we basically repeat the first 3 steps twice. So we ligate on a native barcode, and then we do another NPrep, ligate on a second native barcode, and then ligate on the sequencing adapter or motor protein and sequence. So our construct looks like the bottom one down there where we have our amplicon flanked by basically an inner barcode and then an outer native barcode. So we took our 110 pools of samples and split them into 2, and we native barcoded each of those with native barcodes 1 to 55. Once that was done, we were able to pool each of those into their own pools. We then took one of those pools and ligated it on native barcode 95 into one and native barcode 96 to the other. Once those were ligated on, then we were able to pool those into the other pool. So now we've got pools of pools of pools. We loaded those onto a PromethION flow cell and sequenced. It cost about $1,000 Canadian, so it was a relatively cheap experiment. And we ended up recovering 65 million raw reads, which was actually subpar. Normally we get at least twice that, between 120 and 150 million reads at our amplicon length. So this was actually a pretty poor run, but we used basically an old expired PromethION flow cell because we didn't really know if this was going to work or not. And just to spoil it, that actually was more than enough, even at only sort of half capacity, that was more than enough to get successful results. So this is what our final library construct looked like, and there's a lot to unpack there, but briefly we have our amplicon flanked by our forward and reverse primers in blue, and then the inner native barcode in green, which could be anywhere from 1 to 55, and then the outer native barcode in red, which can be 95 or 96 in our case, and then the sequencing adapters would be on the outside there. And when we look at the raw reads, we were actually kind of surprised to see that exactly what we expected was what happened. We don't see that too much in our R&D lab. So as you can see, going from right to left, you have the amplicon, and then we have our forward primer, and you can see the first half being the gene-specific part of the primer that's conserved, and the second half being the unique tag built into the primer that's variable. And then we have the first native barcode, which is either 1 to 55, so it's kind of variable. And the second native barcode, which is native barcode 95 in this example, which is more conserved. And then a little bit of residual sequencing adapter is also visible there. But basically exactly what we thought might happen actually did happen, so we were happy to see that. We were able to recover at least one read from 96% of these million or so samples, and quite a few of them have a read depth of about 25 to 30 reads, which was more than enough to get a high-quality consensus sequence. So we were really happy to see this, especially on a flow cell that only produced about half the reads we would normally expect. That's as far as we got with the million specimen tests so far, because the data is really big, and it's so big that DNA sequencing is almost now more akin to something like the Large Hadron Collider, where you do a relatively quick experiment and then you end up with just a ton of data that can take months to go through. So we're still analyzing that because it is hard. You can't even open a folder on a computer with 1 million files or open an Excel file with more than 1 million rows. So the data is a bit challenging to work with, and that's just something as a community we're going to have to figure out ways to deal with these large amounts of data these sequencers are now producing. But that's a good thing overall. And then I'll just finish on this one slide to put things in perspective, because I've been talking about DNA barcoding, but really this strategy can be used for a lot of different purposes. So just by using the 96 forward and 96 reverse primers that we have available in our lab, plus the 96 native barcodes that ONT commercially produces, we could theoretically, in our relatively small academic lab, tag over 21 million samples in one library. That means we could sequence, in theory, all 20 million species on Earth for $20,000, or all 8 billion humans for $8 million US, or all 1.5 billion cows for $1.5 million, or all 27 billion chickens for $27 million. Obviously, it's easier said than done, but it's still amazing that theoretically our little academic lab could actually do that in theory. So we're kind of at the dawn of a new era as sequencing output is going to only go up and costs are only going to go down. So I'm really excited to be a part of and to see what new and innovative things are on the horizon, because there's definitely some very amazing things that are coming out pretty soon, I think. And with that, I'll just thank our funders and also thank all of you for coming to the talk.